When researchers look at what we should have in our intestines, you know where they go? One guess. They go to what's inside here, okay? Does that make sense to you? It does in a way, right? Because this has come out of the colon, correct? So it makes sense that if this came out of the colon, if the person was healthy, then maybe we should be putting this back into the colon, right? But that's kind of a flawed argument. You know the reason why? It's because this came out of the colon. This is not food. Give you a little diagram. Recent study looked at fecal transplants. Anyone heard of those? Fecal transplant is where they take a fecal sample from a healthy individual, supposed healthy, and they place it as kind of a suppository transplant into an unhealthy individual. Now, it may seem kind of strange, but in some cases it's helped people. In particular, if they have infections with things like C. diff, quite a few studies have found that it does actually seem to help get rid of that infection. But researchers were questioning, well, what else is transplanted in these fecal transplants? And the study actually found that nearly half of feces is dead bacteria and another form of microorganism very similar to bacteria. We also have some fungus and even viruses, interestingly enough, in the feces. So do you think it would be wise to get fecal transplants? Because we're actually getting dead bacteria, probably some living bacteria, of course, as well, but even dead viruses, or maybe even some active viruses as well. Now, here's one that really would make you wonder if fecal transplants are ethical or desirable. Every feces has a large amount of colon cells within. So your colon actually sheds its lining with every bowel movement, and you have a certain amount of even live, living colon cells, colonocytes, that are in that feces. So should we, because this came out of a healthy individual, start eating colonocytes? See, there's not too much logic to that. Although there may be some healthy species of bacteria here, this food matter has gone through a massive amount of decomposition. And by the time you get it on the other side, after that transit time, that transition period, you don't really want to touch it. You don't really want to bother with it. That's why it's waste. We consider it waste. So, instead of going to the end of the food cycle to get our bacteria advice, let's go to the beginning. Okay, are you with me so far? Let's go to newborn children. Quite an interesting fact about newborn children. When they're born, they have relatively little, if none at all, bacterial flora. But within a very short amount of time, their colons are colonized. If your mother was privileged to be able to breastfeed you, you actually got a boost that put you ahead of your peers in terms of your probiotic load in your digestive tract. Now, if your mother was not able to, there's still hope. We can do things to help the colon even at a later stage in life, but for babies especially, it's so important for them to get that mother's milk. Studies have found, and I alluded to this yesterday, that in the mother's milk there actually have been isolated lactobacilli, certain probiotic organisms living in the mother's milk. And when we consume it as babies, it survives the stomach acid, goes into the small intestine, starts to grow there, and starts to grow there in the colon as well, and colonizes the colon. In fact, most of the microorganisms that colonize the baby's colon are very healthy. They're health-promoting organisms, especially and in particular for those individuals who have been breastfed. Studies have found that most children, newborn and very young age, have larger amounts of two different types of bacterial species, the bifidobacterium and the lactic acid producing bacteria. Maybe the baby has the ideal microbiome during this period of breastfeeding. Does anyone think there's any better food during that time of the baby's life than mother's milk? No, studies have shown that is the optimal food for a baby. And we talked about this last night, that there's actually a chemical found in the mother's breast milk known as oxytocin. And oxytocin actually helps regenerate and make the tissue there that lines the colon much more susceptible to absorption of nutrients. It also makes it more tolerant of toxins. It makes it kind of more stable when exposed to these bacterial toxins that are produced there in the colon. So it's very, very important for babies to get this diet. It's the ideal diet. On the same frame of thought, when we get to adulthood, if we have what we know now as the ideal diet, we will have ensuing the ideal probiotic bacteria growing there. Case in point, what happens when we eat? We chew our food, right? Mix it thoroughly with saliva, and we swallow it. But did you realize there's more incentive for chewing food properly than just absorption of nutrients? Studies have found that there are quite a number of probiotic bacterial species that live in the mouth. This is quite amazing. The study was done in 2000, and it found that 85 lactobacilli were isolated from the human mouth. 85. Among these were L. fermentum, L. plantarum, L. salivarius, and a few others that actually you can go to many health food stores today and buy tablets for these same strains of bacteria, these probiotic bacteria. But they're in the mouths of healthy individuals. Now, when we chew our food correctly, what are we doing? We are mixing those bacteria. They may be in a semi-dormant state, but we're mixing them with something they can use later to eat. They can grow on that medium if we give them the right foods. Before we get to the mouth, we have something that's occurring, and that is dealing with our actual food. You may not realize it, but there are actually bacteria living on these cherries. Does that sound surprising to anyone? Let's take a closer look. What looked like a shiny, slippery surface now reveals there are actually cracks and crevices under the microscope. And indeed, if we look even closer, this is an artist's rendition of what you would see, but you'll actually see bacteria in a semi-dormant state living there in those cracks and crevices. Now, I'm glad we don't have bacteria that look like this. This is like a nightmare bacteria. But it's interesting to note that the food that we eat is what places those bacteria in our mouths. And so when this is all said and done, we are going to isolate some foods that probably are going to be better for implanting those healthier bacteria into our mouth, and some that may be not as healthy to implant other types of bacteria. But to understand the foods and their relationship with the actual bacterial makeup of our mouth and our colon, we need to understand what fermentation is all about. What are they actually doing when they get in an anaerobic environment, meaning without oxygen, which the colon is one such place? What are they doing down there? How are they living? How are they thriving? What are they producing? And so to understand the different types of fermentation is crucial to understanding what probiotics are better and what foods naturally might be rich in these bacteria in a dormant state. For instance, let me give you an example. Has anyone ever made sauerkraut before? What is sauerkraut known for? Raw sauerkraut. As being a probiotic, correct? The reason why is because there are bacteria there that are health promoting, in specific the Lactobacillus plantarum. But it lives there on the cabbage. When we eat this cabbage, we're getting that microbe. It might be in small amounts, it may be in a dormant state, but I'll tell you, once it gets into the stomach, and it actually survives the stomach acid, that strain of bacteria, it gets over into the small intestine, it will start to produce or reproduce. And you'll actually have a probiotic supplementation there because of that raw cabbage. So this is where that bacteria came from. It's quite interesting. So let's look at the first type of fermentation we're going to describe tonight, and that is saccharolytic. Does anyone know what the term saccharose is referring to? Sugar. Okay. So this is referring to the breaking down of sugars. There are different types of sugars that can be broken down. We're going to look at those and the different bacteria that break them down. But this is a broad spectrum of sugar fermentation. Here's a very interesting, very high-powered microscope look at some plant fibers and some bacteria. Truly, they are colonizing this area. But you wouldn't know it by looking at the surface. And a lot of times what we do is we spray vinegar on our fruits and vegetables, right, to clean them. And you know what we're doing? I'll tell you, vinegar is a wonderful antiseptic. It will definitely kill many of these types of bacteria. Did you realize vinegar at the strength of 4% vinegar to water actually will kill Ebola virus? The Zaire Ebola virus. So it's pretty strong. It will kill a lot of these bacteria as well. But if we're looking to get our bacteria from our food sources, that may not be such a good thing because we're killing them with processing them in this way. Another way that maybe this doesn't make it to our plate is if we don't have any raw foods in our diet, because cooking will destroy most of these microbes as well. And also some of the modern methods of food preservation, such as irradiation of fruits and vegetables, you probably are not going to have high counts of probiotic microbes living on your fruit and vegetables if they have been irradiated. So, it's an interesting fact here. It's estimated that 35% of all lactic acid producing bacteria, which are the saccharolytic variety, from raw fruits and vegetables can actually survive the stomach acid, which we talked about last night was the gatekeeper. It kept most of the bad bacteria out and allowed the more probiotic organisms to come through and colonize to a small extent, the small intestine, but to a larger extent, the large intestine. But these fruits and vegetables are rich sources of these probiotic bacteria, and they can actually thrive in the human body. I want to talk first about this bacteria here because this is the kind of the first bacteria that comes into play in our systems as babes. Lactobacilli and the bifidobacterium in terms of what's in the colon in the newborn or the few days old newborn. What is lactic acid, first of all? There have been a lot of research articles that have come out showing that lactic acid is not the reason why we have muscle soreness. Delayed onset muscle soreness is actually caused, and they've realized this now, by microtrauma. You see, when you use your muscles, you actually are causing microscopic tears in those fibers, and that causes inflammation. So it's not the lactic acid. Lactic acid in the body's pH, the solution, turns into lactate very quickly, and lactate is actually potentially very helpful. Case in point, a study that was done on mice a number of years ago found that when mice were put into the maze and allowed to acclimatize themselves to running through this maze, and then they were given a traumatic brain injury, they were kind of dazed and confused. They didn't know how to run through the maze anymore. But another group of mice was fed lactate along with their normal meals. And you know what happened? When they were given the traumatic brain injury, they ran straight through the maze again, showing no residual effects of traumatic brain injury. And so the researchers concluded that lactate is neuroprotective. It protects the brain from injury. Now, these mice were given lactate as a dietary supplement, but where's the primary place that humans get lactate? Any idea? It's through muscular exertion. Because our muscles, when we exercise in higher intensity bursts for short periods of time, we develop lactate in the bloodstream. Because our muscles say, hey, we don't have enough oxygen to generate all the energy needed to accomplish this task, so we will actually ferment sugars. So you have fermentation occurring here in the muscles, but it's lactic acid fermentation. Lactic acid is the byproduct. Other studies have found that the brain actually during exercise, you have channels that open up in the blood-brain barrier that draw lactate in to feed the neurons. It actually is a brain food, if you can think of it in that context. Now, where do you get the lactate from? It's the ideal places from exercise. But there are bacteria that also produce the lactate as well. I told you earlier that Lactobacillus plantarum was isolated from the saliva of healthy individuals. Many of us have Lactobacillus plantarum right there in our saliva. And some people also will take it as a supplement. Studies have found that Lactobacillus plantarum really seems to help to heal the gut when it's gone through some trauma, perhaps some kind of infection or stress or some other issue caused impaired function of the colon wall. Well, this bacteria has been known to go through and exert a healing effect upon the wall of the colon. It primarily produces lactic acid as its byproduct. This was a study that looked at Lactobacillus plantarum supplementation and a high-fat diet plus this probiotic. It was actually two strains, Lactobacillus plantarum and Lactobacillus curvatus. But this was the probiotic right here, so this little diagram follows that weekly administration of the probiotics with every meal. Then we have the high-fat diet plus placebo. Do you think a high-fat diet is healthy, first of all? We learned last night that high-fat diet, or the Western diet, is one of the reasons why we have such problems with leaky gut in this day and age. And we learned also that if we have leaky gut, what else comes out in the circulation? Toxins from the intestines. In particular, lipopolysaccharides, those things that actually batter and beat the brain, and you actually have leaky brain develop because of leaky gut. Much more susceptibility occurs then to toxins entering into the brain. Depression and risk of anxiety goes sky high when levels of lipopolysaccharide, which emanate from the gut, go out into the circulation. So, we really want to bring about a healing response there in the gut and in the brain, but these bacteria are beneficial for more than just that, for more than just healing the gut. They've actually been found to, in this case, reduce levels of cholesterol. That's quite interesting. Even with the same diet, but different bacteria actually equals less plasma cholesterol. Quite interesting, all due to this probiotic organism. I'm not encouraging you to go out and eat a high-fat diet and just take a probiotic to help, because obviously this is still a lot better, right, than this right here. But it does help. It does make a difference. Now here's a pictorial view of what the bacteria did there in the fat cells. They didn't directly do this, but indirectly. This is with the normal diet. You see the fat cells are somewhat small, but over here, the high-fat diet plus placebo, look, they're very large. You know what fat cells do when they get to be too large? They get to be too big, they split, and then they can get large as well, and then they split. So you can have exponential growth of fat cells up to a certain point. Now, do you think most Americans need exponential growth of fat cells? No, we're actually reaching a trend in terms of our obesity levels that's extremely startling. By the year 2025, 50% of the US population is predicted to be obese. So it stands to reason that maybe we could use something like this over here. this probiotic supplementation actually caused many of these fat cells to be almost normal in size. I may not really see the difference here, but on a person's frame, this would be very evident. The fat cells would actually be a lot smaller. Quite amazing, just because of the supplementation of these bacteria. Let's look a little bit more into these lactobacilli. Both of those organisms were lactobacilli. There are, I guess you could put it as good, better, and best, or maybe not so good, better, and best in terms of the whole criteria of lactobacilli. What do I mean by that? Well, let's examine these terms here. Homofermenters and heterofermenters. What is a homofermenter? It's basically an organism that is metabolizing glucose or a primary sugar, a free sugar, into lactic acid. And we learned that the L form of lactic acid is very beneficial, correct? So these are going to be promoting the health in general of the colon and of the body. But there are some other bacteria over here that are heterofermenters. They will take glucose and they'll turn it into 2 parts lactic acid, 1 part ethanol, and 1 part carbon dioxide. Now, how many of you have ever rented a house or been landlord to an individual that has rented a house? Anyone? Have you ever signed a lease agreement? What does that agreement say? It says, I'm not going to change the color of the outside of the house without your permission, basically. I'm not going to restructure the walls or tear down and make a new partition or make some drastic change, correct? Now, when you allow bacteria to go into your colon, you're not signing a lease agreement. You know what? Some of these bacteria are going to be terrible tenants because they're going to change things. And they're going to change things in ways you don't want them to be changed. For instance, studies have found that when ethanol comes in contact with the tight, closed junctions there of the cells that line the colon, it splits it apart. Splits apart the tight junctions. In fact, if they want to try and reproduce colitis or an inflamed colon or leaky gut in the animal model, they'll usually inject it with one of 2 things. Either an enema of ethanol alcohol, with water of course, not so much that it's going to completely destroy all the tissue, but cause at least somewhat of leakiness or the tissue destruction there. Usually it's about 4 to 5% ethanol and about maybe 3 to 4% acetic acid, which is vinegar. So sure enough, when those 2 things are injected, either one of them, into the colon of the animal model, it actually causes colitis, leaky gut. If we want to have the healthiest lining of the gut, we really don't want to have large amounts of ethanol alcohol produced. Is everyone with me on that? Because we don't want to break apart those cells. We don't want to cause leaky gut. And the same thing with some of the bacteria that will produce larger amounts of acetic acid as well, because those could potentially cause problems in higher amounts. So here's an example of a few of these healthier bacteria, and here's some over here that are not so good to have in larger amounts in the colon because of this principle. These are a lot of common homofermenting bacteria. Have you heard of Lactobacillus acidophilus? That's a very common one. Here's another one, Salivarius. Remember, this one actually has been found living in the saliva. So you may ask, well, where can I buy that? Well, eat plenty of fruits and vegetables and you probably won't even need to buy it because you'll have it there. Lactobacillus lactis, another one. And going over here, I have another category of these probiotic bacteria. Now, these bacteria over here are a little bit different. They do the same function as these bacteria here, but they can take on a different type of sugar that these bacteria can't handle over here. But they can take on other sugars that are less common in terms of the whole realm of sugars, but sometimes need to be dealt with all the same. Now these bacteria over here, they will take the sugar and they'll convert it into the lactic acid, ethanol, and carbon dioxide. So over here we have things like Lactobacillus San Francisco. Has anyone ever heard of that strain of bacteria? Does anyone like sourdough bread? San Francisco sourdough? It has a very distinct taste. It's actually raised or leavened by this bacteria right here. Now, how does this bacteria leaven or raise the bread? I'll give you an interesting story. Lactobacillus plantarum, I took it one day and I tried to make a sourdough starter, and sure enough, it smelled sour, and I kept working with it over a period of a week, and finally I thought I had a big enough supply of this sourdough starter to raise some bread. And so I made this whole loaf of bread. It sure looked nice. Put it in the oven. You know what happened when it came out? Flat bread. It was completely flat, completely unleavened. Why is that? It's because this bacteria did not produce any carbon dioxide. It didn't produce any ethanol. It just produced the lactic acid. It was sour flat bread, basically. But these bacteria over here are very beneficial if you're trying to leaven your bread. But do you want to leaven your gut? That's the question we probably need to ask. Do you want to have large amounts of carbon dioxide and ethanol alcohol there in the gut? Does anyone covet bloating or high amounts of gas? Probably not. And of course, we don't covet having our tight junctions of our epithelial cells that line the colon broken apart by the ethanol either. Now, these bacteria will do that with any type of sugar. So ideally, you want to have a mix of these bacteria here and less of these over here, colonizing your colon. Now, there's some interesting names here. Some of them actually suggest the function, such as what we read here, the San Francisco. I don't even know about this one, Lactobacillus confusus. I don't know if I want to get near that microorganism right there. But some of these actually grow on plants. Interestingly enough, the stomach acid stops their growth. It doesn't allow them to grow very rapidly. This one right here grows on cabbage, along with the Lactobacillus brevis. Once the pH gets to a lower state, such as what's found in not quite a pH of 2, which is found in the stomach, but getting close to it, pH of about 3.8, these stop functioning, and you have the Lactobacillus plantarum that really kicks in and starts to grow more and replicate. So ideally, we want to not necessarily supplement with these, but they're going to come into our food probably from time to time. The key is to have our stomach functioning properly so that it does exert that gatekeeper function to keep those heterofermenters out. Now, hetero, what does that mean again? It means different. Do you want your colon to be different than the way God intended it to be? If not, then you want to go over here to the homofermenting lactobacilli. Okay. If we want to have a healthy production coming from these lactobacilli, what we need is to have a shorter amount of time in which the food is making a transition from the mouth all the way to the bowel movement. It has a lot to do with constipation. Does anyone think constipation is an issue here in the Western world? Most definitely, because a lot of times because of our sedentary lifestyle and our very deficient fiber diets. Because of those two criteria, what happens is our transit time is drawn out very long. So that you could even be taking probiotic bacteria as a supplement. Healthy probiotic bacteria, but you know what? They finish all the good food. Glucose, 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 fructose, fructose, fructose. Then they say, hey, there's none of this left. Let's go to the xylose. And we'll eat that. It's kind of like me when I was a child going to the Brussels sprouts. Didn't really want to have to do it. But if I was staying there at the lunch table long enough, I had to eat it. But these bacteria will start to produce some byproducts over here that you may not necessarily want. So one of the most important things we can do for gut health is to ensure that our colon is functioning properly, that peristalsis, which is the muscular contractions that move food along through this long small intestine and over here into the colon and out through the rectum, that peristalsis is occurring correctly. We have plenty of fiber that allows for this bulking action in the stool so that you can actually have it pushed out of the body so that it doesn't stay there for long periods of time. Studies have found that in North America we have much higher transit time than people that are living in third world countries. Children actually have better transit time than adults, but I'll submit to you that if we're undergoing stress or we have a sedentary job or something like this, we probably don't have the best transit time. What's something that we can do to speed up the movement through the small intestine and the colon? Well, one thing, at least for the sake of the small intestine, is to deal with stress in a proper way. We'll talk more about that in our last presentation because stress actually shuts down the movement there in the small intestine and it promotes overgrowth of bacteria, and these are going to start consuming things that you might not want to have consumed. Otherwise, if the transit time was shorter, you may have never had those byproducts produced. But because the transit time is extended because of constipation or because of stress-induced stoppage of peristalsis or the muscular movements through the colon, then we have problems resulting. So it's very important to take that into consideration. Okay, let's go to another aspect of transit time. This is one aspect of the digestive process that's been overlooked in a lot of modern research. I found a study that was actually published in Dr. Agatha Thrash's notes, very prominent physician who's passed away recently, but was one of the preeminent physicians in terms of gut health, lifestyle medicine, natural remedies, etc. The stomach usually empties itself in about 2 to 4.5 hours, depending on the complexity of the food. Series of tests have been run in which persons have been given routine breakfast consisting of cereal and cream, bread, cooked fruit, and an egg. These stomachs were x-rayed and found to be emptied in usually less than 4.5 hours, which fits into that earlier criteria. A few days later, these same persons were given the same type of breakfast. 2 hours after breakfast, one had an ice cream cone. What's that called? A snack. He was found to have residue in his stomach after 6 hours. So instead of 4.5 hours, this x-ray revealed that there was still something there in his stomach after 6 hours because of that ice cream cone snack. Okay. Another was given a peanut butter sandwich 2 hours after breakfast. He was found to have a residue after 9 hours. That's one peanut butter sandwich. Okay. A third was given a piece of pumpkin pie and a glass of milk 2 hours after breakfast. And he was found to have a large amount of residue after 9 hours. So what is this doing to transit time? What is snacking doing? According to these studies, it's slowing it way down. Why is that? It's because the stomach works something like this. It has a job to do, and once the food settles down here in the stomach after a meal, it knows what it needs to do. It's analyzed the chemical makeup of the food. It secreted the right amount of hydrochloric acid and pepsin, which is the digestive enzyme of the stomach, and it knows just the right amount of churning and the duration of churning that needs to happen before this sphincter, this muscle right here, will fully open up and allow the contents to pass over to the next processing plant, which is the small intestine. But if 2 hours after the stomach has begun its work, we supply a new challenge to it, it has to start all over in its process. Because the small intestine will sample some of this and say, stomach, you're not doing your job. For some reason, we have a lot of undigested food still here. So it tightens that muscle even more and says, okay, go back to work. And so the stomach is kept continually at work with continual snacking. Talk about delaying transit time. This is a perfect example of something that can do this. Now here's one that really amazed me. The fifth subject was given a routine breakfast at 8 a.m., twice during the morning and twice during the afternoon. One bite of chocolate candy was given. At 9:30 p.m., 13 and a half hours after breakfast, more than one half of the morning meal was still in the stomach. At 9:30 p.m., more than one half of the meal was still in the stomach. And that's only a few bites of chocolate candy. That's astounding. So we see that nibbling on something or snacking really does delay the whole transit time. So what does that give opportunity for? It gives opportunity potentially for bacterial overgrowth, and they can produce these byproducts that are not necessarily the most healthy. So I want to encourage you, let not this be the end of all your studies in terms of the whole realm of probiotics, but let it be the beginning.